Hello student, good afternoon. Today we will start the acquired immunity. We have to study the innate or natural immunity and resistance of immunity. Also, this topic is acquired immunity. Recovery from a disease confirms immunity to that particular disease, sometimes for life, sometimes for only a short time. Some diseases are held in check by the defense of the host without being completely eliminated, so that they go into a relatively quiescent chronic state. Example: syphilis, Chagas disease, malaria, amoebiasis, etc. As the long as the parasites remains in the body, the host is protected against reinfection. This conditions is called as premonition. The host and parasites exist together in a more or less delicately balanced state. When the resistance begins to fall off, the parasites multiply sufficiently to renew it, sometimes causing a temporary relapse in the process, but the resistance may not rise sufficiently to eliminate the infections entirely. Such as situation seems to be a very common one in endemic areas, not only among protozoan parasites but among helminths as well. Example: hookworms, schistosomes, etc. Except that in this case multiplication of the parasites is replaced by renewed acquisition of them. The same general principle applies to the immunity to arthropods bites. As long as there is a continual exposure, most individuals are capable of developing and keeping up some degree of immunity. The basis for all acquired immunity. Acquired immunity means to create the immunity directed specifically against a particular parasite is probably an alteration in the structure of certain protein molecules, so that they have an affinity for the antigens that stimulated them. When free in the circulation, these molecules are called antibodies, but they are in the cells that produce them before they get into the circulation and may be there long after the free antibodies are gone. Almost any protein substance may act as an antigen, that is, stimulate antibodies that will react with it. With the exception of the simplest viruses, infectious organisms are far from being simple antigens or even mosaics of a few antigens, but may contain hundreds of the different antigenic substances in the form of enzymes, hormone-like substances, toxins, and other metabolic products, as well as the body proteins. In the course of a disease or after immunization by injection or organisms, the disintegrating dead organisms stimulate such abundant antibodies against a few their body proteins that in a in vitro test these antigen antibodies reactions would completely mask any enzymes anti-enzymes reactions. It would be like grinding up a cat to use an antigen against the cat's cellular and blood proteins would certainly mask any reactions to salivary enzymes, yet knocking out even a single essential enzymes in a parasite might handicap it enough so that it would fall easy prey to the natural defensive mechanisms of the host. This is in fact that we do when we administer antibiotics, or sulfa drugs. It is odd that so little attention has been paid to the possible knocking out of essential enzymes. Knocking out of essential enzymes by development of specific anti-enzymes. Unfortunately, bacteria are not easily dissected mechanically or chemically to get the pure enzymes to experiment with. It is suggestive, however, that a small number of metabolizing organisms living in a host body may be more effective than 1,000 times the number of injected dead organisms. This is where the advantages of metazone parasites come in. They can dissected and different tissues and organs used as antigens, as can their collected metabolic products, and they can be induced to produce immunity under conditions which preclude liberations of their body substance, making it possible to study the immunizing effects of their metabolic products independently. Even with some protozoa, a distinction between these two types of immune reactions can sometimes be made. Taliaferro and Taliaferro demonstrated in 1922 that rats develop a reproduction inhibiting antibodies or ablastin as well as trypanocidal antibodies to Trypanosoma levici, that is the genus and species. The trypanosomes are not injured in any way by the ablastin except that they are unable to reproduce, which in the writer's opinion is due to interference with nutrition and consequent growth. This antibody differs from others is not combining with its supposed antigen. The trypanosomes, when mixed with them, in the writer's opinion, ablastin is probably an antibody which is stimulated by and reacts with metabolic products of the trypanosomes, interfering with its nutrition nutrition but not killing it. If this true, a similar phenomenon might occur in other organisms as well as including bacteria and explain the disappointing result from immunization with dead as compared with living vaccines. Later 1935, the writer found that nematodes Nippostrongylus in the intestine of rats likewise had their nutrition interfered with so that they were unable to grow normally or to reproduce. In this case, it is obvious that this immune reaction could not have been directed against the parasite's body substance but only against enzymes or metabolic products of the parasites. That this is likewise true of the migrating larvae of this worm in the parasite parenteral phase is suggested by Charles in 1938 observation that when the Nippostrongylus larvae are placed in immune serum, precipitates form at the external opening and in the intestine, indicating an immune reaction to metabolic products. And second, by the fact mentioned above that living migrating larvae and far more effective in producing immunity than dead ones, the development of precipitates by living larvae in immune serum has since been demonstrated for many other ailments. Campbell in 1954 demonstrated increased resistance to trichinella in mice after injecting them with excretion and secretion of trichinella larvae. Okay, thank you. Remaining part we will be continuing next period.